the endomembrane system, if you take, you're having this is the cell membrane, isn't it? This is the cell membrane. Then you're having nuclear membrane and endoplasmic reticulum at the down, and this is the Golgi complex. Can you see what I'm writing on the diagram? Endoplasmic reticulum at the bottom and Golgi complex at the middle, and these are the lysosomes which are involved in phagocytosis and endocytosis, exocytosis processes. So all are having membranes around them. So the endomembrane system includes what parts of a cell that I'm going to explain here. So when you take the endomembrane system will have nuclear membrane at the bottom of the endoplasmic reticulum. So cell membrane plus Golgi complex plus endoplasmic reticulum, and you'll have the nuclear membrane also. Along all with this, we'll have lysosomes. So you'll be having all these parts under endomembrane system. The word endomembrane system was introduced by Semello Golgi.
Yes, ma'am, he discovered Golgi bodies.
Yes, Golgi complex. Yeah. So the same person had, yes, Bhumika, yes. The same person had introduced the word endomembrane system to designate all these parts. Okay. This is a kind of concept where a definition of endomembrane system has to be noted down, including cell membrane, nuclear membrane, Golgi complex, endoplasmic reticulum, and lysosomes. So question can be framed like they may give a list of cell organelles and they may ask you which one of is not related to endomembrane system. Suppose if they give nucleus in options and ask you to recognize which is not part of endomembrane system, then you can say nucleus is not part of endomembrane system, but only nuclear membrane is part of endomembrane system. Or they may ask you ribosomes, are they part of endomembrane system? No, they are not part of endomembrane system. So the question can be framed in either way. So Semello Golgi is a scientist who gave the name endomembrane system to all the cell organelles and membranes. Okay, that's what yesterday I just want to show. Now, coming to the microbodies concept. Here, after lysosomes, the next concept is related to microbodies. The name itself indicates micro, very minute cell organelles within the cell are called microbodies. Now, under microbodies, we'll discuss about peroxisomes and glyoxysomes. So, these are the two microbodies present in plant cell and animal cell. How a particular organism can carry such microbodies for what purpose, that we are going to discuss now. First, I'll discuss about peroxisomes. Then we can compare peroxisomes with another microbody called spirosome. So it's something related to animal cell and plant cell difference that I'll tell at the end of the peroxisome explanation. First, we'll see peroxisomes. Okay, microbodies examples are peroxisomes and glyoxysomes. That's all for this slide. Then when you take the peroxisomes will contain a membrane around them and we'll observe them in only eukaryotes, not in prokaryotes. Now the term was given by Rodin, that's a scientist's name. This is just introduction about peroxisome. Okay. Now when you take in detail about peroxisome contains some enzymes like lysosomes. But the enzymes in lysosomes are different from peroxisome enzymes. What is the difference that we are going to see? When you take peroxisomes, they're having similarity in having enzymes. Okay, but what kind of enzymes peroxisomes will carry is oxidase enzymes. So which is different from acid hydrolase enzymes in lysosome. So while discussing the lysosome, I explained how hydrolysis, I think Arnav, you asked what is doing the hydrolysis, isn't it? Breaking of water molecule, I explained, isn't it? So that hydrolysis you will find in lysosomes, whereas the enzymes which are there in peroxisomes are oxidases. The other difference between lysosome and peroxisome is the lysosomes, they are arising from Golgi apparatus. isn't it? But whereas peroxisomes, they will be formed from endoplasmic reticulum. This is a very basic difference between lysosome and peroxisome. When they ask the similarities, we can say lysosomes and peroxisomes both are very minute particles present in eukaryotes only. That's the similarity, isn't it? Lysosomes are also there only in eukaryotes. Peroxisomes are also there only in eukaryotes. You won't find them in prokaryotes. That's the similarity. What is the second similarity? Both the organelles are containing particular enzymes. Now, when it comes for differences, what kind of enzymes are there? In peroxisomes, we'll find oxidases. What are oxidases? I'll explain in the next slide. When we come to lysosome, we'll be having acid hydrolase enzymes, acidic enzymes. Then the second difference between peroxisomes and lysosomes is lysosomes are originated from Golgi apparatus, whereas peroxisomes are produced by endoplasmic reticulum. Okay, so that's a brief introduction about peroxisomes. Now, when you take the term peroxisome, it's something related to hydrogen peroxide, H2O2. I hope you know this molecule. This is a very toxic material to the cell because it acts as oxidizing agent. And such peroxide has to be degraded, which will be carried out by peroxisomes. So degradation of hydrogen peroxide is related to peroxisomes. Whereas lysosomes, they'll degrade cellular debris, foreign material, everything they'll degrade because they're having acid enzymes. But peroxisomes are containing only oxidases enzymes. So they are involved in hydrogen peroxide degradation only. And not only that, they'll synthesize even hydrogen peroxide. How the same organelle is producing and destroying hydrogen peroxide is the interest here about peroxides. Okay. Now, how the peroxide is produced, how it is destroyed, that we are going to see. Now, when you take the peroxisome are having different oxidases enzymes, I said. What these oxidases will do, they'll combine the oxygen and hydrogen to form hydrogen peroxide. So it's a usual process which happens during several cycles. Like you'll be having various cycles inside the cell, like glycolysis, gluconeogenesis, like that. You'll be having various cycles where some substrate, here they have given some substrate. I'm not getting into the terminology of the substrate, but this substrate when reacts with oxygen molecule, along with the product, a byproduct like H2O2 will be formed. The equation would be slightly blurred, but don't bother about the equation because these are the biochemical reactions which are not required over here to understand in depth. The only point over here is how a peroxide is produced. How it is produced, that I am going to explain. So a peroxide will be produced when any molecule interacts with oxygen and a product is formed. A byproduct like hydrogen peroxide may be produced in various processes inside the cell. Now this hydrogen peroxide is toxic to the cell and it has to be removed out of the cell, which will be done by peroxisomes. The specific enzyme is catalase. Catalase is the enzyme which will deplete hydrogen peroxide into non-harmful water molecule and oxygen. See here, this peroxisomes are microbodies. So first point you have to understand. So peroxisome is a microbody. Now the remaining matter, don't bother about the remaining matter, that I'll explain later. Now understand about peroxisome. So peroxisome are similar to lysosome in one way and they differ from lysosome in other way. That I explained already. Now here peroxisome contains different enzymes. The enzymes are called oxidases. So what is the function of peroxisome? That I am explaining over here. See, inside the cell, you'll have various processes, isn't it? Various processes in which a molecule like a substrate will react with oxygen and you may get a product. Along with the product, sometimes in some processes, hydrogen peroxide will be released out. Now when you take this hydrogen peroxide, H2O2, the hydrogen peroxide is toxic to the cell because it is highly reactive and it acts as an oxidizing agent. So this hydrogen peroxide has to be removed from the cell. We can't stop formation of hydrogen peroxide because it is formed as a byproduct in several reactions like this. Any substrate, any product, when they react with oxygen, you may get hydrogen peroxide. So this has to be removed from the cell. How it will be removed from the cell? By the activity of an enzyme called catalase. So catalase is the one which will convert the harmful hydrogen peroxide inside the cell into harmless water molecule and oxygen molecule. So what is this catalase enzyme role? It will make the hydrogen peroxide which is toxic to non-toxic material. That's what happens by catalase. And catalase is the enzyme which will be released by peroxisome. Okay. So here oxidizers are the enzyme which are involved here. So I'm putting an arrow over here. Here they are involved oxidizers. Oxidizers are involved in production of hydrogen peroxide and catalysts are involved in destroying hydrogen peroxide. So both synthesis, nothing but the production, and degradation of hydrogen peroxide is carried out by peroxisomes. That's the function of peroxisome. Is that clear now for you? Any doubt after this? The reaction. So hydrogen peroxide. Okay, fine. Bhumika, yourself, hydrogen peroxide are produced and also degraded by the same cell organelle, peroxisome.